All right, there might be a few people still joining, but I want to welcome everyone. Thank you for joining us for this lecture today. I'm Professor David Hess. I'm a professor in the current chair of biology at Santa Clara University. I'll have my contact information on the first slide when I share my screen. And feel free to jot that down and send me any questions about the talk or the biology department. A little background about myself. So I grew up in Cleveland, Ohio. I went to Case Western Reserve as an undergraduate to study biology. And then I did my graduate work at Harvard Medical School, and where I got my PhD in genetics, studying microorganisms. I then went on and did my postdoc at Princeton University in their Department of Genomics, where I was again studying the evolution of microorganisms and using whole genome sequencing and other genomics techniques to better understand microorganisms, especially in the context of evolution. And for the last 13 years, I've been a professor here at Santa Clara University, where I continue that research on the study of evolution of microorganisms, with a lot of recent focus on the public health. So looking at things like the spread and evolution of antibiotic resistance and the spread of bacterial pathogens. All right, so I will share my screen. Okay, let me know in the chat if there's any problems seeing this screen, or actually just unmute yourself and jump in. But hopefully this is fine. So today I'll be telling you about, and so there's some context for this. This is sort of adapting how I set up a lecture in my upper division molecular biology course. This is taken by almost all of our pre-med students take upper division molecular biology. It's required for the biochemistry major. We have bioengineers, neuroscientists, public health science majors. Most of our life sciences majors try to take this course before they graduate. And so today I'll be exploring the sort of mystery in the molecular clock. I'll explain what that is on the next slide, and how this provides key insights into the mismatch repair system. So hopefully most of you have seen sort of a picture of the tree of life, or a similar picture before. And if you can see on my, hopefully you can see my cursor here. Whenever you have these branch points, so this branch point down here is the point between vertebrates and invertebrates. And we timed that. We know that obviously there was a time where invertebrates split off from each other in terms of lineages, but we can time that and get this approximate 700 million years ago by using the molecular clock. And we do this by comparing genomes. So when you compare genomes from each side of that split, at some point they had to share a common ancestor long ago. So you look at those key fundamental genes that all living creatures have, and you start looking at mutation rates. And what we've learned since we started sequencing DNA in the late 70s is that mutations occur at a pretty regular frequency when you look over evolutionary time. And so figuring out the sort of ticking of that clock, so the ticks are the accumulation of mutations. If you know what rate that's occurring, then you can compare any two genomes of different species, or even two members within a species, and you can figure out how long it's been since they shared a common ancestor. And so this is a really useful tool in evolutionary biology. And this has been used to sort of build the entire tree of life and the relatedness of all life on Earth. But what came out when this technique was used was an oddity, and I'd like to sort of describe that here. So in theory, 1 out of every 3 mutations, of the possible mutations you could get, could happen by C to T mutations. And I'll be explaining that for the next few minutes. And we'll see on the next slide that though what's really observed is far out of line with that. How could one mutation, though, even, the base case that I'm stating here, how could that occur for a third of all mutations? Don't worry too much about the nomenclature here, but geneticists split up mutation. So this is anytime one of the letters in your DNA change. And DNA has A, T, G, and C. So there's 4 letters. So anytime those change to something else, it can either be a transition or a transversion mutation. And so the transition would be a C to T, a T to C, A to G, or G to A. So there's 4 of those. And then all of the other letter switches you can imagine, you can write this out if you don't believe me, there's 8 other, and none of those could be caused by a CDT mutation. So 4 out of 12 is 1/3. Well, okay, I can understand if you tell me it's a C to T mutation, how does that, how that works, but how does a G to A mutation be caused by a C to T? So some of you might already be thinking about this. DNA has 2 strands. And so when we're reporting sequences, we're just looking at the top strand, but there's a bottom strand there too, and that could mutate. And G binds across from C, and A binds across from T. So if on the bottom strand, a C mutated to a T, on the strand that we're reading, the top strand, that would create a G to an A change. So you can see there how a G to the A change could actually be explained by a C to a T change on the bottom. And so how are the reverses also explained? A T changing to a C, that seems like the opposite, and the same in A to a G. Well, whenever we're looking at mutation, we can only determine if mutations happened by comparing 2 genomes. We have the genome, say our genome we're looking at, and then we're comparing it to some other genome, and we call that the reference genome. And so you're looking for a change, a difference between those 2. Well, if I'm comparing myself to, say, sequence someone else that's on this live stream, your genome wasn't frozen in time. Since we shared a common ancestor maybe 20,000 years ago, we had a great-great-great-great-great-grandmother in common. Both of our lineages have been evolving. So if you pick up a C to T mutation, but I compare my genome to you, it'll look like a T to a C, because that's just the way you compare everything with respect to the reference genome. But in reality, you actually picked up the mutation. And so that's how you get those 4. And then again, you could always pick up that mutation on the bottom strand of your DNA. So that's how you get that 1/3 of the possible mutations that you could see could be caused by a C to T. So why am I mentioning this at all? Well, all of this DNA sequencing that we've done over the last 40 years has shown a very striking pattern. That in reality, so the theory says 1/3 of mutations could be explained by a C to T. But when we actually sequence and compare organisms, we see that 65 to 80%, so nearly 2 to 3 times higher than we would expect, are explained by C to T in the real world when we actually sequence genomes. So something is happening here. Whenever you see a result like that in biology, you get excited because there's something to uncover there. And so we're going to spend the rest of the talk figuring out why this happens. So there's 2 ways that a C to T mutation can occur. The first is in DNA replication. DNA polymerase comes along and it's going to copy your DNA, and it can make a mistake when it's copying. So I'm going to show you a quick movie of this. So this is DNA being replicated. Now, we don't have cameras that are small enough to actually film this, or microscopes that are powerful enough. But we know the structural models of all the proteins involved. We know the structure of DNA. So this is a simulation of what it looks like. And while it may seem like it's going very fast, this is slowed down 1,000 times. So this is in 1,000 times slow motion. So DNA is coming in here on the left. It's going through all of this protein machinery, and you have 2 strands of DNA coming out on the other side. And this is what DNA replication looks like in all cells. A lot of moving parts there. And so in reality, so that was showing DNA replication at about 1 base pair per second. So 1 letter copied every second. But in reality, you're copying your DNA at 1,000 base pairs a second. So it's incredibly fast. And all of that protein machinery that's doing that work it makes a mistake every once in a while. And it makes a mistake about 1 in every million base pairs. Maybe that doesn't sound too bad, but we need to dig into that number. So is 1 in a million base pair error rate a problem? Well, you have 6 billion base pairs in your genome. So that means every time a human genome was copied, DNA polymerase makes about 6,000 mistakes. That's a problem. You have 50 trillion cells in your body. Most of them divide at least once a month. Some of them divide as often as every 2 days. So that would be like your skin, your gut, cells that make up your cornea. If you have 50 trillion cells dividing, and every time they divide, you get 6,000 mistakes in each cell, each division, you would start piling up mutations really, really fast at a dangerous rate. And the reason we care about mutations is the accumulation of mutations over time is what leads to cancer. So if we didn't fix these mistakes, you would be riddled with cancer before you were ever born. So clearly this isn't what's happening. There has to be some way to account for this. And that's where DNA repair comes in. And specifically for replication errors, it's something called mismatch repair. So we're looking at several different structures on the right here of DNA. And so DNA has this nice, this is what you're probably used to seeing is this double helix. This is looking down the barrel of the helix, so you can see it's a really tightly packed structure. This is what the space-filling molecules would look like. And again, this is space-filling molecules from the side. You can see this helix is really tightly packed. There's not sort of empty space like you might look when you see the simple ribbon model. And here we see the hydrogen bonding that occurs. So the letters of the DNA are on the inside of the helix. So G bonds with C, A bonds with T. And the other letters can't hydrogen bond with each other. And so when I make a replication error and I'd put, instead of a C here, I'd put a T across from this G, they can't hydrogen bond. And when things bond, they get drawn closer together. But in this case, if they can't bond, they're going to be pushed apart. And that's going to create a bulge in the DNA helix. And that bulge is really easy for the cell to recognize. And so it's constantly looking for these bulges in DNA, and it knows it has to fix it when it detects one of those. Now, there's two possibilities from the cell's point of view when it detects a bulge. The first is replication has come through and made an error, and in which case the bases will look normal on each side. There'll be a G and it'll be across from a T. They can't hydrogen bond with each other, but they look like normal bases. And that would be the hallmark of a replication error. The other type of error is DNA damage. So this is UV light hitting you, smoking, asbestos, really overly cooked barbecue. So that char tastes good on barbecue, but it's also full of carcinogens. So any carcinogen that would damage your DNA, it takes a normal DNA base and it rearranges the bonds. And a G would no longer look like a G. It would look like something completely different. And it wouldn't look like a C, a T, or an A. It just looks completely different. So if the cell sees a bulge and it sees a sort of foreign base, it knows one of its bases were damaged. That's easy. Cut that out, fill it back in with normal base pairs and fix the damage. But with mismatch, we have when we're looking at replication errors, we have one additional problem. And that is, which base pair do we fix? They both look normal. So I'm going to have to take one of them out and copy the information from the other strand. But if I take the wrong base out, I'm not going to fix the replication error. So the cell has this clever system, and we'll talk about the cell cycle on the next slide, that in G1, before it replicates the DNA, it marks the DNA strands so that in replication, it's going to know which is the old strand and which was the newly synthesized strand. So here's the cell cycle, and I'm going to focus just on the first two phases of the cell cycle. Cells spend almost all of their time in G1. That's where they're waiting to replicate, waiting to get those signals that everything is set for replication. And it's during this phase that the cell lays down these marks, and it lays down methyl groups specifically on cytosines. And it doesn't methylate every single cytosine in the DNA, but 1 out of about every two or 300 gets marked. And then when it moves to S phase, this is when we actually replicate the DNA. After replication, each of the DNA strands will have one old strand and one new strand that was copied from it. And so now if there's a bulge there, and they're both normal bases on either side, the mismatch repair machinery can do this really clever thing. It looks around for the nearest methylated cytosine, and then it can trace back to the bulge, trace the strand back, and it now knows, oh, that strand that had the methyl cytosine 200 base pairs away, that's the old strand, and that's the correct strand. The mistake must be on the newly synthesized strand, because that's the one that DNA polymerase would have just made in S phase. So it knows that's the mistake, and it's going to then use mismatch repair to fix that. And if we had the sort of, if I was going through the full hour lecture, this is when we would really go into detail into each one of these steps and the mechanism. I'm going to just briefly go over that for the purpose of this. So these are these proteins up here that are looking for these bulges, and they're also going to determine which is the old and the new strand. And then depending on what type of species you are, how exactly you cut the DNA out, there's 2 alternate pathways for cutting them out. But what's important is you're going to remove that damage. So on either side here, we see we've removed some of the damaged strain, or in this case, the strain that had the incorrect nucleotide incorporated. And then you are going to fill this in with a specialized DNA polymerase that comes in and fills in this gap, and then it leaves this one little bond behind. We fix that with a process called ligation. And so at the end of that, that fixes it. And this process is amazingly efficient. So of those 6,000 mutations our cells pick up every replication, all of them are fixed. Every once in a while one gets through, but it's very rare. This is an amazing system in terms of its fidelity, and it doesn't really make mistakes. So it takes care of all of those mutations in our body. But that doesn't explain where all these C to T mutations are coming from. And if you remember back a few slides, I said there's 2 ways you could get C to T mutations. One is replication. Well, all of those are going to be fixed here. So what's the second? And the second is through a chemical process called spontaneous deamination. So if a UV light ray hits a methylated cytosine, so not a normal cytosine, but a methylated cytosine at just the right angle, it triggers this chemical reaction. And this chemical reaction converts that methyl cytosine into a thymine. So what happens now? Now you had a methyl cytosine, it's become a T. And that means a T is across from a G, because C would normally bond with G, and that's going to create a bulge. But when the cell looks at this, both bases look normal. So even though this has been generated through damage, the cell doesn't know it. It looks like a normal T. There's nothing that looks damaged about it. And so the cell says, oh, we need to use mismatch repair to fix this. And if we think about what would happen in G1, where the cells spend most of their time, both strands are methylated. So mismatch repair, it's just a dumb set of proteins. It does what it does. It needs to look around and determine which is the old strand. But both strands are methylated. So it takes the first strand it finds is methylated, and it calls that the old strand. Whether that will be the strand that has the methyl cytosine that's been mutated to a T, or the G, is a 50-50 chance. So there's a 50% chance that mismatch repair will fix this correctly, and there's a 50% chance that it'll fix it wrong, and it will actually cause the C to T mutation to come in. So this is this little leak that's in mismatch repair. All of our other, and thankfully this is a very, very rare event. It is very rare that a UV light hits one of our methylated cytosines at exactly the right angle. But when it does happen, there's only a 50% chance that it gets fixed correctly. And there's a 50% chance that it leads to mutation. There's no other event that happens in your body this frequently that can't be repaired. And this is why when we look over time, CDT mutations far swamp out any other type of mutation that you can accumulate. We use that to set the molecular clock. It's that regular accumulation of CDT mutations over time that makes the molecular clock useful. It also, though, is what leads to the accumulation of mutations in us over time. And if you would look at something like a cancer cell, you would see that a lot of the mutations that were accumulated in there were caused by these CDT mutations. So this sort of brings us full circle, shows the link between the molecular clock and this idea, and how that ties into this fundamental repair pathway of how we fix all of these DNA synthesis mutations that occur, but it has this little leak on the back end where these mutations that occur in G1 are still going to slip through the station. So with that, I'm going to stop the share and we'll open it up to questions. And these can be about the talk or anything about the biology department. So you can either type them in the chat and I'll try to get them, or you can unmute and I'm happy to actually see a person and hear their question. All right, well, thank you for letting me know that the lecture was interesting. It is, I have to also say, it is very strange to be lecture sitting down. I wish I was there in person with you. Not being able to sort of lecture back and forth and right on the board is very foreign. So this is also kind of a strange format. Absolutely. So if you are interested in veterinary medicine, all right, questions are rolling in quickly here. So try to go through these. So yes, I've actually had several students take this course. So this is going to be material that's foundational for any of your professional exams, like the MCAT, GRE, any of those that go that you need to take for any sort of pre-health profession. And most of those courses, you will take molecular biology in medical school and vet school, nursing school, physician assistant. And I've had many students come back and say that, you know, they've used their notes and that taking my course at Santa Clara really helped them. They got an A when they took it and they felt they were so far ahead of their other students, like in medical school or veterinary school. Did you ever take more than one chemistry? Oh, I took all the way through physical chemistry. So I took a full year of Gen Chem, a full year of OChem, and a full year of PChem when I was an undergraduate. So I graduated with a ton of chemistry. You don't need that much chemistry at Santa Clara, but our biology majors do take a full year of intro chem and a full year of OChem to get the degree. What's the ratio between time spent between lectures and lab? Good question. So we've actually recently redone our intro series in the biology. So it used to be 5 courses and 2 of them had labs. It's now 3 courses and all of those have labs. So in that intro series, you're spending, I would say about half of your effort is spent in lab, half of it is in lecture. And then when you're getting into the upper division courses, you need to take 7 more. 5 of those have to be with labs. Lecture is about 3 hours a week and lab is about 3 hours a week. So a biology major is a very lab heavy course. I would say you spend somewhere between, of your biology courses, probably about 40% of your time is spent in lab. It's something we're really proud of here. Our students, a lot of our students go directly into biotechnology, into the biotech industry in Silicon Valley with our degree. And we've got great feedback from employers that they really come out with practical laboratory skills and more training than most undergraduates receive. In the mismatch repair process, it looks like a large number of base pairs. Yeah. So it turns out it's really hard for a big protein polymerase to come in and add just one base pair. So whenever the cell is removing, it removes a stretch of damage, and it moves about 10 base pairs. And that creates sort of a landing pad where that polymerase can come in and then fill in that. And that's just sort of the stereochemistry and the steric hindrance of just fitting a big protein complex in. It just turns out it's easier if you have a stretch of DNA. So that was a good observation. How strong would I say? Our best students that come through at biology, we've had students go to all the best top 20 medical schools, PhD students in programs like Harvard, Stanford, University of Washington. And again, like I said, we have placed many of our graduates directly into jobs in biotechnology. I don't know off the top of my head what our rank, and I'm actually not aware of any sort of rankings by department. But it is not like the biology department is weak relative to the ranking of the other departments. And if you're looking at this, you've probably heard the new Sobrato campus is launching next fall. We're very excited to get into this. It's going to be a real state-of-the-art laboratory facilities. I mean, the whole building is going to be amazing, but we're really excited to get into these new lab facilities. What does undergraduate look like in the biology department? We have a lot of it. Almost all of our faculty are research active. Obviously, with coronavirus, it's been a little bit weird. We've been finding ways to do remote things. A few students are back doing research. We really hope that next year or our next quarter, winter quarter, we should be back to having more labs that are more full. I have 3 research students with me right now in lab. There are labs that have as many as 7 research students in them. Most students that really want to, that are motivated, get in to research labs. If you just like, oh, if I apply to one place and then they're full and then you don't look again, maybe you won't get into a lab. But we really try to provide for students that want that research. And I would say of my research students, 2/3 of them have been pre-med. So there was asking about like, does that sort of fit with pre-med? It does look really good on the applications. And I would say about 2/3 of the research students are pre-med or pre-health of some type. Are most of these upper division more discussion-based or lecture-based? Lecture-based, again, with that strong lab component. How much do you interaction with professors? So again, this is one of the things that's great about Santa Clara. TAs do not teach our labs. So for this class, you're going to have me 3 hours a week in class, but you're going to have me I will teach you the 3-hour lab. And that's really where we get to know our students' lab, because there's always some downtime in lab, like a gel's running, you're waiting for a reaction to occur. And we've got 12 to 16 students in a lab. And so I'm just talking with the students and interacting them. And I find in those lab environments is where I get really close personal connections with the students. I really get to learn what their lives are about. There's opportunities to do mentoring there. That's one of my favorite things about being a professor here. What if you're interested in the biology field but don't want to go pre-med? So I've talked about that some. So going research for a PhD, going into just directly into biotech. We also have an agreement with the Keck Graduate Institute, KGI, which is down in LA. They have a really interesting program, master's program, that's half a master's in biology and half an MBA that's focused on the business of biotech. We send about 1 or 2 graduates a year into that program. There's preferential admission if you do something called the biotechnology minor at Santa Clara. And the students that go into that kind of go into venture capital. They work on some of the marketing or some of the business side of biotech. And we've had graduates that have started at Santa Clara for their biology degree, then went to KGI. They have great careers with great companies, and that's another type of option. And then there's another thing that I always like to put in a plug for is, we're constantly hearing about testing with coronavirus right now. Well, a lot of that falls on the backs of our public health labs. And there are great careers in public health science. One of my main collaborators, he right now is the state lab director. So he runs all of the public health labs in the state of Nevada. He's actually been named their testing czar. And we do a lot of research together. I've been helping him, not on coronavirus, but using whole genome sequencing to identify bacterial infections and to track antibiotic resistance in the state of Nevada. We need brilliant minds getting interested in public health science. We can see the need of it with the pandemic that's going on right now. And that's another career option that I think is often underexplored. Would I be able to take this and other science classes, even if I weren't majoring in hard science? So my molecular biology class, you have to complete the entire intro series. So that's 3 lower division biology classes before you can take this. So this isn't something you could just, this particular class you couldn't just dip into. But the biology department does teach several non-majors courses. So you need no biology prerequisites to take them. And those all do have lab components as well. Do AP Bio, they do not. So you get credit for them, but it's just generic credit. So that'll give you better registration times, help you graduate faster. But all of our students have to start in the Biology 1A, the first class of the intro series. What do biology labs look like during COVID? Different. So we're offering our first in-person labs again next quarter, but spring quarter and fall quarter, everything has been online. So we've used a lot of lab simulation. All the data analysis is the same. So we still do data analysis. A lot of it is focused on interacting with primary literature. So it's become, we've altered it to be more sort of research focused. And one of the things that we're doing is, because everybody's learning how to deal with this on the fly, we're going to have some really small, like standalone 1-week modules where if students that come out of their intro series during COVID don't feel comfortable with like pipetting, solution making, doing a type of certain sort of field biology, there's going to be an opportunity once we're kind of through the pandemic to take these 1-week refresher courses to bone up on those skills before they enter the upper division labs. So that's one of the ways that we've thought about dealing with these crazy virtual learning situations that we've been in. Okay, mismatch repair bases from each other. No, so it's actually good when you have mismatch. So you wouldn't want to force base pairing between mismatched base pairs. Otherwise, you wouldn't know you have to fix it. So it turns out that it's actually good that the mismatched base pairs don't properly bond. If you were to give us a pop-up test on the lecture, what would be one of your main questions? Sure. So one of my favorite questions that I've given and this now brings in the other repair pathways as well. I give students an example stretch of DNA, and then I give them a set of conditions under which that DNA is exposed to UV light. Then they have to look at that sequence and predict which repair pathways would be recruited to that damaged piece of DNA and then justify their answer. So they've got to look through and see, okay, given this sequence and these conditions, how do I expect this DNA to get damaged? Is it in one way? Is it in multiple ways? And then what repair mechanisms from the cell would come in. So that's a really way to show that you understand the material deeply, that you're synthesizing it. So that's a type of question that I would ask. That would be a higher level question from this material. Is there any ways for mismatched DNA base pairs that are from? No. So that is, so once, so that's the scary thing. Once a mutated base pair has been incorrectly fixed, it is a permanent change. Now, a caveat that I'm going to give there, and this isn't really a caveat, but it can be not passed on to the next generation, right? So it would only be on one of your chromosomes. So you don't pass both of your chromosomes on to each of your children. You pass one of them. And so, you know, that's one of the sort of ideas. You can learn more about this in our evolution class. If a chromosome picks up a bunch of really deleterious mutations, that chromosome will be selected against over time. So evolution, so it's evolution is the one that's coming in, and over time, if you get a chromosome that builds up a bunch of mutations, it selects that out. But there's no like molecular mechanism inside. Once a mutation is fixed, it's permanent, and only evolution can take care of it then. Is there a difference between a BA and BS? No, both have advantages and disadvantages. It's often what you sort of, most of the time, bachelor's of science degrees take more classes that are science focused. If you have a bachelor's of arts degree, you might have an ability to add like a different minor or maybe even double major. So it's just your sort of package for medical school just looks different with a BA and BS, but students get in to medical school with both degrees at, I would say, at pretty equal frequency. How to office, so for most of my upper division classes, I have review sessions before the test and then office hours by appointment. So again, we have the lab classes. A lot of times students just ask me questions during the lab when we have downtime. They're always asking questions after class. We just step out in the hallway, and I normally am doing 10 minutes of sort of impromptu office hours after almost every class. And then if somebody's really stuck, then they just meet with me in my office, and however long it takes till they understand the material, that's how long it goes. Are you doing any research? What does it look like during the pandemic? So I am doing some bioinformatics research. I'm kind of doing more paper writing and grant writing right now. And the other reason is because of my background in epidemiology, I've been tasked by the university. I'm actually heading our tracking and tracing unit. So for the past 9 months, I've basically been, helping stand up a small public health lab or a public health office in our at our university, and that's taken a tremendous amount of time. So I've sort of stepped away from research a little to do that. But what I did do over the summer with my research students is we learned Python coding, and we've been writing some bioinformatics scripts and using them to study some whole genome sequencing we have of E. coli isolates and comparing commensally, which are non-disease causing that we get from our intestines, to E. coli that have been purified from urinary tract infections from students on campus, and seeing what differences we can see between those disease-causing strains of E. coli and commensal strains of E. coli. So we've been analyzing that. Average number of hours for, it's about 10 hours a week during the school year. And it can be up to full-time over the summer. Is it hard for freshmen to find research experience? Harder, but I've had freshmen in my labs before. I would say the typical starting year is sophomore year, but definitely if you are really gung-ho to do research, I bet you could find someone's lab to work in as a freshman. How many students are in the average 1A class? So our new intro classes have the sort of flipped classroom design. So it's about 45 to 50 students. you're working in groups. So there'll be some amount of lecture, and then you'll start working on a problem, analyzing some data, setting up an experiment. And then we have and they're team taught. So we have multiple professors, and then they're going around to the different tables to help you work through the problem to see what you're doing. And then we'll come back and lecture for a little bit. So that's been running for about 3 years now. And I think it's been really successful. What do I teach other than molecular biology? So I teach bioinformatics, and I teach the biology 1B, so the second one, which is focused on DNA and evolution. I've also taught project lab, and I would like to teach a sort of biochemistry. And biochemistry teaches one, but I'd like to teach one that's focused on sort of metabolism and more the biology of biochemistry than the chemistry of biochemistry at some point. And what would you say to students in STEM? What would you say that students in STEM fields, biochemistry, are more collaborative? Oh, absolutely. So I've had biochemists in my lab, bioengineers in my lab, public health sciences in my lab. There's a lot of professional organizations. So, things like, there's a healthcare ethics program that a lot of different life science majors go through. A lot of them are also EMTs on campus. There's a lot of areas where the students are mixing, and they take so many of the same classes from the biology and chemistry and physics department that it's collaborative. And I think with us all moving into the same big building next fall, it's going to be even more collaborative. So I think I got through all the questions there. I apologize if I missed over one. I'm trying to keep up with the chat. And actually that's basically time here. I will share in the chat my email address again, just in case anyone wants to jot it down. Feel free to email me any questions. And thank you for showing up, and enjoy the rest of the open house events that you'll be attending today. And hopefully see some of you next fall.